Good morning, all. Today we will be learning about the basics of DNA. The objectives of learning this is to understand the nature of DNA, the composition of DNA, and the structural difference between purines and pyrimidines. We all know that DNA is double-stranded, and it is composed of a ribose sugar, that is deoxyribose sugar, a phosphate, and the bases, that is purines and pyrimidines. So the sugar and phosphate always makes the backbone of the DNA. Here in the image itself, we can see that alternating phosphate and sugar is going to be making the backbone of the DNA, and within these two strands are the bases that are stacked one upon each other. So adenine always pairs with thymine with a double bond, and the bond is hydrogen bond here. And guanine pairs with cytosine, and these other triple hydrogen bonds that we see here. Along with that, in this structure, we can also see that there is presence of minor groove and a major groove. Now, there is a specific difference which we can notably understand here, that the major groove is having the sequences which are specific for certain proteins to come and bind, whereas minor groove does not have any specific sequence, and so a non-specific protein binding occurs in minor groove. Coming to the chemical structure of this DNA, this is how a sugar and phosphate is going to be making up the backbone, and the bond formation that takes place here is phosphodiester bond. And if we see adenine and guanine, these two are going to be the purines, and thymine and cytosine are the pyrimidines. So we all know that why it is important to study DNA, because from DNA, it is if it's a gene, it is going to get transcribed into an RNA, and from RNA, this mRNA is further going to get translated into a protein. So a functional protein is going to be formed, which is considered to be a workhouse of so many processes. Now, when we look into this DNA and RNA, when we look into the name that has been given to DNA, that is deoxyribonucleic acid, we need to understand why is it named so. So when I call it to be deoxyribose, what does it mean? So we know that the sugar that is present in DNA is a pentose sugar, that we call it to be a ribose sugar. But then this ribose sugar is having a less oxygen at second position, that is here. So we can see that the 2' of this sugar is not having hydroxyl group here. So we call it to be a 2-deoxyribose sugar, and so we call DNA to be deoxyribonucleic acid. Okay, so nucleic acid with an intention, because it is always going to be seen in the nucleus, and acid because the phosphate which is there in the backbone, it contributes to the presence of acidic molecule within the DNA. So this is the major difference between RNA and DNA. So in DNA, when we see the sugar that is present, it is always 2-deoxyribose sugar, and in this RNA, which we call it to be ribonucleic acid, it is going to be having the hydroxyl group to be present at the second position. So this is one specific difference which is there. Along with that, we also know that there is in place of thymine in your RNA, we have this uracil. So when we look into uracil and thymine structure, there is one difference. Then again, you have to remember that the difference is in having the methyl group. So in thymine, there is a presence of methyl group, whereas in uracil, methyl group is not present. So uracil is present in RNA, and thymine is present in DNA. Now, coming to the configuration of this DNA, when we see, so as already been spoken, so these two strands which are there, they are going to be composed of repeating units of sugar and phosphate. And inside we have these stacks of these particular pairs. That is, A is always going to be pairing with T, and G always is going to be pairing with C, that is cytosine. So if we see there is a specific distance between these tags, and it is 0.34 nanometer. The turn with which this DNA, so one turn which we see is having 3.6 nanometer, and the diameter of DNA, if we see, it is 2 nanometer. So this is how the structure of DNA looks like. Again, I'm emphasizing here that the DNA always runs in 5' to 3' direction in one strand, and the other strand will be exactly opposite, that is 3' to 5'. So where exactly this DNA is found? So DNA, when we talk of eukaryotes, in eukaryotes, there is a nucleus, where in nucleus, the DNA content will be there. But along with that, in mitochondria also the DNA is there. And in plants, if we see, in chloroplast, the DNA is there. Coming to virus is either going to be having DNA or RNA. Never both will be present. Coming to this bacterial structure, in bacteria, they do not have a true nucleus, and so the bacterial chromosome is always said to be lying naked in the cytoplasm. Along with that, the bacteria is also having a plasmid, which is said to be an extra chromosomal material. All right. So now let us see the structural difference which is there between this purine and pyrimidine. So by looking at the structure itself, we can make out that the purines that are adenine and guanine, they are double ring structures, unlike the pyrimidines. So pyrimidines, when I see, they are the bases which are going to be single ring structure. Now let us come to understand what is the individual difference between adenine and guanine itself. So when I talk about adenine, we have to understand and we have to memorize what exactly it stands for. So adenine, if we see, it is called as 6-aminopurine. So when I call it to be 6-aminopurine, what does it mean? That at the 6th position, there is a presence of NH2. And so it is said to be 6-aminopurine. So if we remember the name, this name itself, it will be very easy for us to memorize the structure as well. Coming to guanine, so guanine is different from adenine, as it is 2-amino-6-oxypurine. So here we can see at the second position, there is NH2 which is present. And at the 6th position, if we see, there is oxygen present here. Okay, so hence guanine is 2-amino-6-oxypurine, and adenine is 6-aminopurine. Now let us come to pyrimidine. So pyrimidine are simple structures which are single ring structures. Now thymine, if we see, thymine is said to be 5-methyl-2,4-dioxypyrimidine. So if we see at 5th position, there is presence of methyl group. Okay, and at 2nd and 4th position, there is presence of oxygen. So it is 5-methyl-2,4-dioxypyrimidine, right? So this is your thymine. Now, when we see cytosine, cytosine is 2-oxo-4-aminopyrimidine. Sorry. So it is 2-oxo-4-aminopyrimidine. So this is like at the 2nd position, if we see, there is oxygen. At the 4th position, if we see, there is NH2 present. And so the name has been given to cytosine. Coming to uracil, it has already been said that the difference between thymine and uracil is in uracil, the methyl group is not present here, right? So what exactly it is going to be called then? So it is simply 2,4-dioxypyrimidine. So you can see that if the name change, if we see, only this 5-methyl is missing from uracil, and so it is 2,4-dioxypyrimidine. So this is the difference between purines and pyrimidines, and also individual difference between themselves itself, like so how adenine differs from guanine and how these three differ from each other in the structure. Okay, so what exactly we have learned? If you are able to answer this, then I hope you have understood it very well. So what exactly is the DNA? Like if there is DNA, the DNA has dash sugar in its backbone. So you should be able to know what kind of sugar is present. Then the second comes, what are the names of purines and pyrimidines? Then how many bonds are there between, how many hydrogen bonds are there between guanine and cytosine? And this one is like a very simple one, but still in eukaryotes, if I ask, in eukaryotes, the DNA molecule is present only in nucleus. So whether it is true or false. And the last one is the phosphate sugar bond, which is present. It is what exactly? So I hope you all have understood this. And thank you. That's it.